Good morning and welcome to the third session of the JGI user meeting. I am Nikos Kirpidis and I will be your host for this action-packed session with an outstanding list of speakers, starting with Matt Sullivan. We all know we are living on a microbial world. No offense to my friend Dan Roxer. And we all know that we have more microbes on Earth than there are stars in the universe. Yet, how much do we really know about viruses? We know that there are at least 10 times as many as the microbial cells, with a total number of about 1 followed by 31 zeros. Or by using the goals of Carl Zimmer, if we'll stack them together, we'll reach 100 million light years. And still, we know very little about those. In this war against our ignorance for the most dominant life form on Earth, Matt Sullivan is no stranger. In fact, he has made it his mission to change our view and our understanding and knowledge about them forever. So, and from what I have heard from him yesterday, we are all in for an early morning treat. So, Matt.
Thank you, Nikos, for that very kind introduction, and thank you for the opportunity to speak. I really do want to just try to emphasize that the goal of my lab is to try to bring these tiny entities into light. The goal is to bring this incredible unknown into every organism's viewpoint, because from microbes up to the biggest organisms like whales on the planet, all of them are probably heavily impacted by viruses. So I just want to start out, because I'll go right up to the end, by thanking the people who do the work, in particular 3 postdocs, Lee Deng, Jennifer Brum, Karin Homfeld, and 2 graduate students, Cesar Ignacio Espinoza and Bonnie Hurwitz. I'm focusing specifically on their work. The other members in the lab shown here have contributed greatly to these projects, but these are the leads of the stories I'll tell you today. So to try to map out where we're going to go, first I just want to introduce why you should hopefully care about viruses. Then I want to actually get into this big unknown. 90% of everything we look at is unknown. And I'll tell you that we're trying to attack this unknown problem in 3 ways. The first is using model systems. I came from 10 years of work with cyanobacterial viruses, or cyanophages, and a postdoc, Karin Homfeld, has brought a totally new model system to me, and I'll tell you about some unpublished and really exciting aspects of her work. The second vignette is just some methods. We're attacking some of these problems with novel and actually retro methods. Quantitative TEM is a method that revives some 1970s electron microscopy methods to actually look at the particles, and it's unveiled a very interesting aspect of viral biology I'd like to share with you. And then viral tagging, which is a very new cutting-edge method, which revives some 10-year-old thinking and just puts it in some higher throughput context. And finally, I want to wrap up with what I think are some novel and interesting approaches to just bin and organize this unknown sequence space, and then informatically try to walk through what kinds of things this unknown sequence space could be doing. So I realized yesterday, as I talked to a lot of people, I should probably not just start with why viruses, but why microbes. Nikos just mentioned they're very abundant. In the oceans, microbes make up 98% of the ocean biomass. They're absolutely critical to all of the biogeochemistry that fuels the oceans and Earth. They control production and consumption of organic matter and oxygen. When we think about greenhouse gases, many of those are produced or manipulated by microbes. The entire nitrogen cycle, every step is regulated by different microbial players. And then in many different gradients that we look at with respect to pH and redox, it's usually microbes dominating those changes in chemistry. And maybe most apt for this talk, microbes have an enormous potential for different microbial traits, different functions that they can actually sustain for maintaining metabolic flexibility across many different environments. And I always quote Dave Carl, because he just says, microbes make things happen, and I think it's just that simple. So why ocean viruses? There are 10 to the 31 viruses on the planet. In the oceans, there's about 1 million to 100 million viruses per milliliter of seawater. That's about the only thing we know very well. That is that we can count viruses pretty well by looking at DNA stains. Viruses are probably responsible to between 10 and 50% of microbial mortality per day. They also move genes around, horizontal gene transfer, or HGT, from one microbial host to another, and they impact the evolutionary trajectory of microbes in a very profound way, and I'll tell you one story about that. They impact biogeochemistry both just by lysing these microbes and changing fluxes of nutrients, but also by the particular genes that they might move around can often have big biogeochemical impacts. And then when you think of toxins as a human, Bordetella toxin, cholera toxin, Shiga toxin, pertussis toxin, these are actually all encoded by viruses integrated in a microbe. They're not really microbial toxins. And so it's when that virus gets a little out of whack that those particular toxic epidemics can manifest themselves. This particular picture here that shows tiny little green dots is microbial cells and then viruses as the really tiny pinpricks. And what hopefully you'll see is that there's about 10 times more viruses than cells. And we know that these are not just big numbers, they're very dynamic. In the oceans, these viral populations turn over on the scale of every few days. So we've got a very abundant, dynamic population of viruses out there. But what we actually don't know when we look at a slide that's been DNA stained like this is whether this virus infects this host or this virus infects that host. We have no idea whom they're infecting or what their genetic repertoire is. This is a talk for the DOE, so I thought I should also mention the big gun. By far, the largest flux of carbon on the planet is through viral lysis. This is some work that Curtis Suttle summarized for the Earth system, and you can see the numbers are a couple of gigatons per year for most of these fluxes. At the ocean surface, about 90 gigatons per year, and viral lysis is responsible for about 150 gigatons per year of carbon flux. The problem is everything is unknown. These are pie charts where the orange is telling you from 4 different ocean regions where they have viral metagenome sequenced, the Sargasso Sea, the Gulf of Mexico, British Columbia, and the Arctic Ocean. About 90% of what we look at sequence-wise is completely unknown, no hits to anything in any databases, not even environmental databases. So the challenge is to understand that unknown. And again, we take a model systems approach. The first model system that I'm interested in are the ocean cyanobacteria, Prochlorococcus and Synechococcus. They're ecological champions. They're very, very tiny, less than a micron in size, but they're the most abundant photosynthetic cells on the planet, about 10 to the 5th cells per mil over the entire oceans. They're responsible for something like 15 to 30% of carbon that's fixed on the planet. Because of those reasons, the JGI sequenced back in the days when a phage genome was a relatively decent-sized JGI project, 3 phage genomes. And those early stories basically were that we had 3 types. So for cyanobacterial viruses, there's 3 types, myo, podo, and cyphoviruses. And these are very consistent with what people had seen for E. coli and sewage, and yet these are viruses from the open oceans. And the real surprise was when we looked at the genomes, okay, there were genes that looked like they were viral, but there were also genes which looked like they were bacterial. And at the time, this was a really, really big and interesting story, and it led to about 8 more years of research summarized here. Basically, they had core photosynthesis genes, PSBA and PSBD. These are expressed during infection. They can actually, when you do molecular evolutionary studies, they can generate diversity not only in the virus, but when they place subsections of that gene back into the host. So they're generating diversity for these hosts. If you look in microbial metagenomes for some of these genes, it turns out that about 60% of those you can identify are actually viral. We can tell something mechanistically about why they're there. It turns out that host range and the length of time that a virus is in the cell controls whether they have a PSBA or PSBD gene. And modelers can actually show that this is important to the phage. The virus has stolen this core photosynthesis gene for its own benefit. It actually helps the virus increase its fitness. And then I'll just allude to the fact that I've touched on photosynthesis genes, but there are a number of other auxiliary metabolic genes or host genes that these viruses have stolen, probably for their own end. And in each case, it's probably to alleviate a bottleneck of some critical metabolic step that the virus needs to increase its fitness and productivity. And just to bring it from the tiny virus entity back to the global scale, this is a happy Prochlorococcus cell floating around in the ocean, 10 to the 5th per mil, vast regions of the oceans. The virus here actually has genes which interrogate Prochlorococcus' ability to photosynthesize right at the core of the photosystem, genes involved in stored versus respired carbon here, transalylase, which is sort of a switch between the glycolytic and pentose phosphate pathways, and genes involved in scavenging nucleotides. So these viruses have cleverly tweaked host metabolism to their own ends, and because these Prochlorococcus are dominant in the global oceans, this influences global biogeochemical cycles of carbon and oxygen. And we've now shown in some other systems also of phosphorus, nitrogen, and sulfur. And CO, PN, and S is really all oceanographers care about when you get down to it. So we were excited to be able to show them ways that viruses interact at all 5 levels of those elements. So exploring this unknown space beyond cyanophages, that was sort of a review. In contrast to my favorite host, Karin, a postdoc in my lab, loves Bacteroidetes. And you should too, not only because they're in a lot of places in the world, but also yesterday Michael Schartz mentioned that when microbes get out of balance in a mouse, it turns them from fat to lean. Well, those microbes that are actually the important balancing act are Bacteroidetes. So in the human gut, a large chunk of your microbes are Bacteroidetes. They're also important in the marine system. Up to 10 to 40% of cells in any marine environment by fish are Bacteroidetes. And by metagenomics, it's about 10 to 30% of the sequences that we can map are Bacteroidetes. So these are a widespread and abundant phylum. And Karin spent her PhD, before she came to my lab, isolating a bunch of viruses off of her favorite little homeland in Sweden. And she came to my lab, and we've now sequenced 30 of these genomes and created 13 proteomes. And I was honestly very excited about Karin when she came to the lab, but I basically thought it was the conservative, boring project. Cyanophages, there are 3 types, myo, poto, cypho. Karin's phages surely will be 3 types. Well, she proved me completely wrong. There are 13 types, at least, in this group of viruses. We recognize them this way. Basically, you can think these are pairwise comparisons of the fraction of the genome shared. If they're dark, that means they share some genes. If it's white, they don't share a single gene. So there are clearly 13 sets of boxes here. So based on genomes and whether you share a gene or not, there are 13 groups. 4 different groups of podoviruses, 1 group of myoviruses, a number of groups of cyphoviruses, and 2 groups of single-stranded DNA viruses. I was completely blown away by this. New model system, totally new diversity. Well, these are really interesting because if we go to NCBI and download every viral genome, by myoviruses, Karin's, so in the box plots are those viral genome sizes by NCBI, and in the stars are those viral genome sizes from Karin's model system. And you can see the myoviruses are maybe they're not so interesting. But the podoviruses are bigger by far than any podovirus that's been seen. In fact, her biggest one is twice the size of the largest podovirus. As well, in the microviruses or the single-stranded DNA viruses, she has some which double or triple the largest microviruses. So by genome size, they're really interesting. And when we dig deeper into these are just to focus on not even some of her biggest podoviruses. They look nearly identical, okay, by microscopy. But it turns out they have incredibly different genomes. This is group 2 viruses, and we define the groups now based on their genome. And what you can see is when we plot a genome, the lines between them are syntenic or homologs. And you can see that the lines are relatively parallel, and most of the genes are shared in group 2. But between group 2 and group 1, those genes that are shared are a handful over here, and they're less than around 20% identical at the amino acid level. And they form a totally separate group. So nearly identical morphologies and completely different genomes. And we've now done a lot of physiological work. They have very different host range stories. There's a specialist generalist story here. The growth dynamics, how long they're in the cell, how many viruses are produced per cell. All of these things are really novel and different between the groups. So these genomes are sort of opening a Pandora's box of important ecological and evolutionary fitness traits that we can now tease into with our model systems approach. We've looked for these in the wild, and it turns out, while they're not very abundant in any sample, they're really common in a lot of them. So this totally new set of podovirus groups are probably ubiquitous, but low abundance, and that's probably why they've never been seen before. The single-stranded DNA viruses, this is hot off the press like last week. These are really big, and we basically spent about 6 months trying to figure out a way to get at their genomes. And Karen had a very clever strategy in the end where she figured out how to do it during infection and basically plasmid prepping them straight out of the cells as a double-stranded intermediate. This clever step worked, and we have these large single-stranded DNA viruses and small single-stranded DNA viruses, and indeed, the genomes are totally novel. So this is a genome of the large one, and a white box means we don't really know anything, no hit. A blue box here is an e to the -3 hit to NR. So anyone who's been doing that for a lysis gene, you should have a much, much better hit. This is incredibly poor, but we'll take it because it's the best hit we got. And then these gray boxes are actually hits to a Bacteroidetes microbe that was sequenced. And so we actually think that this virus has the capability of integrating into a microbe. So that's incredibly exciting for us phage nerds, but we like that. The small one has the same story. Nothing is recognizable except for a very weak sequence similarity to this lysis gene and to a DNA replication gene. So this is totally unknown. In fact, these are so unknown that I said to Karen, I said, let's make sure that these things we think are genomes are really in the viral particles. So she actually used PCR and sequencing to say, yes, these really come from cesium chloride purified viral particles. So we now know these totally unknown things, which are a little too large for the paradigm of single-stranded DNA viruses, are in the viral particles. And we're currently doing experimental proteomics to identify the structural proteins and mapping these to wild metagenomes. So Karen's work and our other model system work, we can now use these models to try to identify other unknown genes. And the first way we can do that is with whole genome transcriptomics and things like biolog plates to look at functions of particular cloned and expressed genes that look interesting. This allows us to get at things like nutrient stress genes, regulators, et cetera. Variant structural proteomics I've hinted at a couple times. This is incredibly powerful for us. All we do is make a clean cesium chloride prep and send it to our mass spec collaborator, Nathan Verburkos, and we routinely get 10 to 50 new proteins that are actually annotatable as structural. We did this already on a community metaproteome, and we got 398 new proteins that were annotated now as structural. So really opening up this unknown sequence space and helping us understand what some of its function might be. We can refine those functional assignments in collaboration with Hua Chu at Baylor College of Medicine to look at cryo-electron microscopy and really detailed structure of these viruses. And that allows us to place unknown spots of electron density and try to make hypotheses about particular structural functional assignments. And I always tease my lab, one of these days we have to break down and do some genetics. We don't have good genetics in these model systems yet, but we're just that sort of bright genetic-enabled student or postdoc away from doing that. So if you're doing genetics and want to pioneer a new model system, please give us a thought. Okay, beyond model system work, we've been developing these new methods. The first one, again, is retrobiology. This is Jennifer Brum and an undergraduate, Ryan Schenk, who are using quantitative electron microscopy to get beyond these dots to these beautiful images. They're basically just taking pictures of every single virus they see, and they're doing it in a quantitative manner using 1970s technology. This is taking an airfuge and just spinning everything down onto a grid quantitatively. And this has been incredibly powerful. They've looked at hundreds of samples now and thousands of viruses. I want to just share one brief story from their work because it's relevant to the sequence world. Here what we've done is simply plotted morphology-based estimates of different viral types, or quantitative TEM, against metagenome-based estimates. And what we see is that cyphoviruses, really using either method, probably about 5% maybe of the community. Myoviruses in red, using either method, still on the one-to-one line-ish, around 10%, 15%. Podoviruses, we're way underestimating based on metagenomics. And this is probably reflective of the fact that Karen goes out and tells me about 2 entirely new podovirus groups. We're probably got a lot of work to do in podoviruses. And then the one I was really surprised by is non-tailed viruses. These are the things that are probably going to be single-stranded DNA, RNA viruses. Look like they fall on a one-to-one line with all the no-hit metagenomic sequence data. That implies that the vast majority of the unknown sequence space in metagenomes might not be myopodocypho at all. And we might be chasing the wrong members of the viral community. So that's very exciting for me because I don't know much about these kinds of viruses and I love to learn. So this is a new way to go. We like to link viruses to their hosts. In particular, I've always been bothered by fragment recruitment in metagenomics. Particularly microbial metagenomics, if we have a genome that's along this axis and we map wild metagenomic reads to it, you get plots like this, which go from, say, 50 to 100% nucleotide identity along the genome. And obviously, okay, 100% identity, you might believe that that metagenomic fragment belongs to that organism. But what about the ones that are 90% or 95% or 50% or 30%? And we don't have a good quantitative metric. And so we've been working hard to try to develop a population genome scale understanding, locus by locus. What should the sequence identity be to believe that that metagenomic fragment maps realistically, scientifically, rigorously to a reference genome? And we do that in two ways. The first is just brute force, and I'll tell you about that in a second. And the second is developing what is a relatively simple method. Phil Hugenholtz came up to me at a meeting and said, Matt, why don't you just take these viruses and stain them and then use the virus as a tag? And I said, oh, that's a good idea, Phil. Can I do it? Because he had too much work to do. So he said, yeah. So we did it. And the concept is you can label your cells, and this is to look at 10 to the 4th cells that are examined per minute using an Amnis image stream flow cytometer, which takes a picture of every particle which goes through your flow cytometer. And what you can see is that we have cells that are unlabeled in the control experiments, and these are red because they're cyanobacteria, so they autofluoresce red when struck with blue light. And those are complemented by viral tagged cells. The viruses that we label with cyber get stained green, and so those red cells now have a little green pinprick. And this is exciting because then on a flow cytometer, you get cells which are not tagged down here in yellow, and you get cells which are tagged with your viruses in blue. And if you could separate populations in a flow cytometer, you can do large-scale population genomic screening, which means we can start to ask whom infects whom on a very grand scale. So we applied this off of Monterey, California, where we actually looked at linking viruses to their hosts using Synechococcus 7803. And we did it in two ways. The brute force way is underway with some sequencing here at JGI, where we isolated 75 phage genomes at each of 2 sites. And this is about 3 years of work to get those isolates. So this is a tremendous effort. It's also a tremendous effort on the sequencing side and the library prep side. In contrast, we could do viral tag metagenomics, where we actually go out and we sequence the community in blue, which tells us maybe a couple of % are cyanophage, and the viral tag population in red, which tells us maybe 60% of the population is cyanophage. So clearly, we've enriched viral tagged wild viruses for these particular viruses, which are host specific, from about 2 to 60%. And so that's at least a 30-fold enrichment. And I'll argue, as we refine our understanding of these, the other 40% are also cyanophage. It's just a database bias issue. So what does this mean? It means that it changes our understanding of a viral species concept. So this is a population genome landscape. This is just each new genome compared to a reference panel of genomes to create a vector that we can input into a PCA analysis. And what it shows us is that 4 genomes that we isolated are separated, and then the 3rd and 4th are nearly on top of each other. In fact, they were so on top of each other, they were 99% identical. And I said, oh no, we sequenced the same thing twice. I was wrong, because about 3 SNPs is what we should expect per genome, and there were about 300 SNPs. So 100-fold greater than sequencing error is the variability. So this made me think, ooh, these could be from the same population. Maybe we've just defined what a population is. And indeed, when we do the viral tagging, we can pull out, and this is one viral tagging experiment, all these different populations. And we know they're populations now because we can subsample. The orange dot is the average, and the scatter of information around it is the variability in the metagenomic reads recruited to that. These are different populations whose clouds do not overlap. This implies genetically isolated material. So, and then the third aspect that I wanted to bring up is that if I use any host in any ocean environment, I get the gray dots. So these are all isolated cyanophage genomes that I spent 10 years isolating, and that's their variability. So what we learned from this simple 10-minute experiment and subsequent informatic work, of course, are 6 classic ecology and evolutionary biology lessons. First, we can tell you something about the number of viral populations, probably more than 20, infect this particular host. Second, their intra-population variation is less than 5%. So if I go back to fragment recruitment, I'm not going to trust anything that's greater than 5% variable, at least for this type of virus in the ocean. Third, the interpopulation variability is actually rather large. One population to another are at least 20% divergent, and even those genes that are shared are around 20% divergent. Fourth, because we have the metagenomic reads, we can actually quantify who is the most abundant population, and does that change over space and time? So this is now getting into ecology. And the fifth is sort of a classic tax area relationship question. It looks to me like local diversity is greater than global diversity. And this has been hypothesized in the viral literature a couple of times with very little data. The star there is to remind me that all the gray dots are culture-based, and these other dots are not. So I need to worry about the culture bias issue. And sixth, back to the ecology again, we now, I think, have a very deep understanding of all the possible host genes that are in these cynophage populations from the wild, again, from one experiment. So we'll just briefly go through some informatic approaches that we've been taking. Again, everything's unknown. If you wanted to answer two basic questions in viral metagenomics, who is there and what are they doing? About 5 to 13 or 15% of your data could you talk about? So again, everything is unknown. So we just went back to Shibu Youssef's work, my favorite paper of the original Gauss series. And sorry, Doug. Only because it allows to create bins for unknown sequence. He basically said, let's use sequence-based similarity to create protein clusters. The concept is, you've got unorganized metagenomic fragments, let's at least organize them into bins. And this is really powerful for us. We increased the amount of protein clusters that were known 10% with the first viral metagenome we looked at. Viruses don't share a single gene, so even asking a question, which environment is more diverse than another, is pretty much an impossible question. But this protein cluster data allows us to ask that question at the level of the community. I can tell you that DCM communities off these Tara ocean samples, deep chlorophyll max communities, are more diverse than surface communities. And this is using protein clusters as in straightforward rarefaction analyses to look at diversity of the viral community. We're also interested in how this gets us towards sort of the global ocean virome to help us hone in on what particular genes are most important to all ocean viruses. First, we're just going to focus, though, on 32 Pacific Ocean viral samples that represent many surface samples over space and time and many deep samples, heart of the ocean, oxygen minimum zone kind of samples. Real diverse collection of samples, and this represents both the first quantitative viral metagenomes out there, and I'd be happy to talk with people about why we think these are the first quantitative viral metagenomes, and some data from two different CSPs that I've had sequenced here at JGI. Not surprisingly, most of the reads are unknown, but we can start to organize this unknown sequence space. 6,454 reads, 456,000 protein clusters, 27,000 of which have 20 or more members, which is sort of the gold standard for something we trust. And just to give you some context, this doubles the known viral sequence diversity out there. So just organizing this sequence space. We can begin to ask questions about sort of the kinds of proteins that occur in one environment or another in this Venn diagram here. Or what I'm going to focus on right now is just the core flexible idea. I think from 32 ocean viral metagenomes, we've now saturated the pan or flexible genome. It's probably around 420,000 different proteins that exist in ocean viral communities. And after only 2 or 3 viral metagenomes, we probably got at the core. So there's about 180 core protein clusters, which are critical and abundant to all these samples. This is totally new data last week, and dissecting the core is, we're really trying to understand from these data sets now which ones make ocean viruses tick. And the exciting thing is that, again, even these things that are shared across all the oceans can be completely unknown, and they would be ideal targets for sort of high throughput structural and synthetic biology experiments. But I'll just tell you what we do know. In the 180 that are shared by every sample, most of the signal really is myo, podo, and SIFO. And I alluded earlier that that's not true when you look at every protein cluster. And what we think is going on is that myo, podo, SIFO are everywhere, but they're not very abundant. And it sort of hit us in the face that, yeah, duh, we get that now. And this is really the case. They're everywhere. They make up the bulk of what is shared across every sample, but they're not very abundant. If we look just in the photic zone at what's unique in that photic zone, most of that information is unknown, but the stuff that's known is very focused on these photosynthesis genes and other cyanophage core genes I've mentioned earlier, like PSPA, PSPD, plastocyanin, ferredoxin. There's also some incredibly new stuff coming out, so iron-sulfur clusters that are involved in nitrogen fixation, as well as other aspects of respiration, which we haven't delved into much. New flavors of carbon metabolism. They're getting into the pentose phosphate shunt in ways that are well beyond what just the cyanophages do, as well as succinate metabolism. And then if we look in the aphotic zone, or the deep oceans, even more of these core protein clusters are unknown. And there's a couple of interesting stories. The first is that prophages, or integrating viruses, are probably more important in the deep ocean. And they seem to encode for maturation of flagella, which implies that motility of their hosts might be a unique niche-defining feature for these viruses to get to survive in the deep oceans. And that's just the stuff that's shared by everything I just alluded to. There's 1,870 niche-defining protein clusters based on the statistical analysis we've done, where, that might define ways that viruses are adapting to a particular host and environment. For example, nearly the complete TCA cycle is represented in these data sets, including some shortcut mechanisms akin to the PEBS story we've already published. And then in nutrient-limited open ocean samples, there's clear signals of phosphorus and iron uptake stress proteins. So really exciting for getting at their ecology. I think we've got, with this organization structure, some clever ways to annotate these things informatically. The first is we take advantage of SciMap, which, as far as I can tell, is the ultimate unknowns database. They've pooled every single aspect of information out there. The second is we've developed a method we call ecological profiling, where we use feature selection algorithms to select from all the very, very, very rich metadata we have for these protein clusters to help us understand whether, say, proteins cluster 2 and 3 might have similar functions, and whether we can ever link cluster 3, for example, to a known functional protein. And then finally, we're using co-occurrence and network analyses and some new annotation tools at PhageRest to try to get an understanding for when co-occurrence of different protein clusters occur. Does that help us understand function, create hypotheses that we could test at the bench? So in summary, I hope you've learned that viruses obtain these host genes or auxiliary metabolic genes to adapt to new hosts and new environments. And my lab in particular is using 3 ways to explore this unknown sequence space which dominates our viral world. The model systems, where we actually try to do classic bench work to understand in low throughput and high throughput ways the functions. Experimental methods such as quantitative TEM, which point us towards the right viruses to look at, and viral tagging, which help us understand en masse how viruses are linked to particular hosts. And then the ways we're trying to use clever strategies to link this information informatically and try to develop hypotheses which are testable. There's a tremendous number of collaborators and folks who I'd like to thank, in particular the Tucson Marine Phage Lab members who I've thanked along the way with slides. And I'd like to thank the Department of Energy for longtime support through CSPs, both mine and my old advisors, Penny Chisholm, and the Moore Foundation, Bio5, and Biosphere 2, who took a flyer on a young kid and really helped me get the resources I needed to develop a lot of these stories in a very big way. And the work that I detailed in the North Pacific is funded by the National Science Foundation. Thank you for your time. Steve?
Questions?
A lot of viruses are isolated by looking for plaques or looking for devastating damage to the host cells. And yet, in nature, a lot of viruses don't kill their host cells. So I'm wondering whether you think that that many of these viruses could be brought into culture if more sensitive assays for infection were applied.
Yeah, great question. Absolutely, you could get a bigger diversity of viruses if you went beyond lytic. I think we're so far from saturating the lytic world that that's a good place to start. At the same time, I didn't talk about, we've developed a method we call phage fish, which actually looks at viral copies intracellularly. And we think it's the first assay to allow you to do that second thing you'd like to do. So to allow you to look at an integrated viral infection in a cell at the single cell level and be able to tell whether it's going lytic or not. So I think you're right. New approaches could really help us getting at the lysogenic or the temperate phages. And we don't have good methods yet.
Yeah, my second question is about, it's widely believed that viruses are the source of a lot of the variation in hypervariable regions of microbial genomes. And yet, I haven't so far seen evidence that really convinces me that viruses are the whole story. One would think with all the viral metagenome sequencing going on that there would be a convergence on an answer to that question. So where are we in that process?
Well, we haven't looked at that particularly, but I'm just thinking I need to do a fragment recruitment plot with viral metagenomes versus microbial metagenomes from the same sample to start to think about that. And you might suggest that there would be a statistical overrepresentation in the viral metagenomes in the microbial islands, and that might be a good step. I don't have any other clever steps in mind, though.
My question is related to what Steve was talking about with the lytic cycle versus the temperate phages. And I'm just wondering about the functional genes that you find. And do you think that they're always related to supporting a lytic phase? Is that why they're interrogating these pathways?
No, they're probably not always related to a lytic pathway. I didn't mention that, so the viruses that we're talking about that are not lytic actually have a lytic pathway as well. So the temperate viruses can be either integrated or go lytic. And I didn't talk about the fact that about half the phages we mentioned in this talk are actually probably temperate, and we haven't shown that definitively yet. I think that you could imagine host genes that are adaptive both during a lytic infection and during a temperate phage infection. So, and those might have a slightly different flavor. And in fact, some of the best work was from the, I believe it was 1977, Lin Chow's work that showed that, one of the key host genes that these viruses had was actually more important during lysogeny. So it's a new, very unexplored realm. Neat to think about.
So when you're calling them host genes, I'm not as familiar with this as I should be probably, but are those from homologous to the actual Synechococcus host, or are they host-like and they come from bacteria in general?
Yes and yes. In the case of the photosynthetic genes, if you use a bunch of different techniques, they will very clearly have come from their hosts. In the case of transadelase Tal-C, for example, they look like they come from some proteobacteria as opposed to cyanobacteria. And there's a couple of cases like that. Most of the cases are from their known hosts. And we don't have, I'd actually love to talk more about that because I think we don't have a good understanding for why that might be or is.
Hi, you showed that nice slide that had some very distinct sequence clusters of viri. And I was wondering if you could tell us anything about the role of recombination in generating those, whether there appears to be one or if you have any ideas.
So relative rates of recombination that help constrain those clouds? Is that kind of what you're after, or?
Whether recombination is shuffling diversity among them, or if it's pulling the clouds together, homogenizing them?
We definitely haven't analyzed those data in that way. We just published a paper in EM on T4 phages where we carefully looked at core gene sets. And amongst those core gene sets, there very much was a sort of phylogenetic cohesion, if you will, that came out of the intragenic recombination that we could observe. Now, admittedly, that is not always detectable at very, very close phylogenetic distances. But given the sensitivity of the tools, it looks like probably intragenic recombination, at least, is happening much, much more often within closely related phylogenetic groupings.
With apologies, I'm going to butcher the acronym, but and in fact, I won't even try to translate it, but these elements called CRISPRs
Yes.
that are seen in a certain number of virus bacterial systems. Do you see any of them here?
Yeah. Great question. Never in cyanobacteria. And I have no idea why. Yeah. Those CRISPRs are elements which are essentially a roadmap, if you will, or a historical record of past viral infections, where the microbe actually has incorporated a piece of the viral DNA to allow you to, basically as a defense for this particular mechanism associated with these genetic elements. And we don't see them in cyanobacteria. We haven't looked for them as carefully as we should in our viral metagenomes where we have paired microbial metagenomes, because that would be a neat place to explore CRISPR questions, and we should think about that.
I'm sure there are many more questions, but we need to move forward. But still, I cannot resist from asking. You mentioned 500,000 clusters, protein clusters from phage genomes, doubling the amount of what we knew. So I was wondering, what is the number of genes that do not go into those clusters, phage genes that do not go into any of those clusters that you have identified from the complete phage genomes? So what is the amount of singletons, basically?
So the 456,000 number is 2+ members. There's about 30 or 40% more singletons.
So one third more.
Yeah.
So, okay. Thank you, Matt.